Yes, so thanks for inviting me, first of all. And my name is Zilu. Currently, I'm a postdoc researcher at Jesper Olsen's group at this Novo Nordisk Foundation Center for Protein Research. So we are a group of people that are very interested in high throughput quantitative mass spectrometry-based proteomics. And we also have a focus on cell signaling events by quantifying the proteome and especially the phosphorylation events during in all kinds of cells. Yes. So I did glycoproteomics during my PhD. So it was glycosylation itself. It's a post-translational modification. So it is something glycans are something attached to proteins, and they are really important, but normally ignored. I will just give one example, like the SARS-CoV-2, the coronavirus. So now everyone knows the spike proteins on the virus, but not too many people know that on this specific spike protein, it has a lot of glycans on it. And actually we don't know too much about the glycans on it. So if we study more about the glycosylation, the glycans on those proteins, I think we will have a much better understanding of a lot of biological processes. Yes, first of all, I have to say I do agree that it is the best device for single-cell proteomics. And before the cellenONE time, we were thinking how to do single-cell proteomics, but there was no perfect, I mean, it's not perfect yet, but there was no very well-established method for that. But I think cellenONE is really great at the cell isolation and buffer dispensing. And I think the most important thing in single-cell proteomics is to reduce the sample cost. And that's exactly what cellenONE can offer us. Yes, so I think this is a very good question. And as we had a lot of discussions today and in the whole field, so we need to compare proteomics and transcriptomics. So for the proteomics people, we need to think what unique things can proteomics offer. So I think there are two aspects. The first one, proteomes are slightly different from the transcriptomes. So that's why we do need to look into the proteomes. Another unique feature that only proteomics can offer us is actually related to the glycoproteomics. So only proteomics approach can look into the post-translational modifications like phosphorylation and glycosylation. So that is something we think we need to address in the coming future.